Okay, this is a long lecture on diabetes with the emphasis on advanced glycation end products. So I'm going to cover some new stuff, especially on complications related to advanced glycation end products. I think that's going to be rather interesting and new to you. I'm also going to show some of my old diabetes slides. A lot of people are interested in that. They want me to talk more about it. So hopefully this will be interesting for you. Okay, we'll start out we briefly had talked about low protein diets potentially having more benefits than was previously realized. When you don't have a lot of protein around, the liver will burn, more fat will be burned, lipolysis, to provide the glycerol skeletons of a triglyceride that's like a propane triol, and that will be used by the liver to make glucose. So you're going to lead to burning more fat. So a low protein diet gets you to burn more fat, which makes you skinny. And James R. Mitchell, his research, the Harvard guy, who he really believes that increased longevity is not just caloric restriction, it's also protein restriction. So that's an interesting point. And like I said, too, that kind of conflicts a little bit with eat more beans. So we'll get back to that in a future lecture, but that's an interesting point. Kempner diet also is going to have less insulin-like growth factor, meaning less leucine also because of the lack of animal protein, less mTOR. So you're going to have less leucine to activate mTOR, meaning that you're going to have less cell replication, mitosis, and implies less risk of cancer occurring or spreading. Okay, we're going to get more into the regular diabetes stuff here. So this is really about the most important thing you can know about diabetes. It comes from this paper here by Anthony J. James Hamilton, 2020. And the deal was, how does the fat get into the skeletal muscle? And it appears what's happening is that the dietary fatty acids from the blood can then intercalate to the plasma membrane of the skeletal muscle, whereby they can become protonated. They're typically deprotonated in the aqueous solution, but they can become protonated as they interact with the phospholipid bilayer outer face. And then once they're inside that phospholipid bilayer, they can flip-flop to the inner leaflet of the plasma membrane and subsequently enter the cytoplasm. And they can do this in such large amounts that they just overwhelm the skeletal muscle when there's high dietary fatty acids in the blood. And that's why it just depends on what you eat. They tried inhibiting all the plasma membrane fatty acid transporters, but it didn't matter. That didn't really significantly affect the rate at which the fatty acids were entering the skeletal muscle. So it was not carrier-dependent transport. It was simply just going across the plasma membrane in this research paper, and this guy's been studying this for many years. So that's a big deal. So what does that mean? That means that the more fat you eat, the more fat gets into your skeletal muscle, and the more likely you're going to develop insulin resistance and reversal of electron transport, which is a metabolic disaster. The cell cannot control fatty acid entry, which indicates that we're not designed to eat that much fat. And by the way, that's super important. So what it's saying is a diabetic really should minimize their fat intake. Conceptual stages of diabetes, fat. It's basically a progression of where the fat goes. When you get too much fat in the skeletal muscle, that's the first detectable finding of insulin resistance. Then I'm going to show a picture of this and it'll make more sense. Too much fat in the liver, then you start getting fasting hyperglycemia. Initially with fat in the muscle, it's postprandial hyperglycemia after eating. You start accumulating fat in the pancreas. It damages the pancreas beta cells. You get beta cell burnout. Then it starts to accumulate in the arteries in conjunction with atherosclerosis, and you have this whole microvasculopathy. Goes to the eyes, retinopathy, to the kidneys, nephropathy. So this is all the fat aspect of it. The advanced glycation end products are going to be doing damage all over the place too. We're going to come to that in just a moment. Okay, here's the picture. And one can see that the fatty acid accumulating in the skeletal muscle is going to cause, after eating, postprandial hyperglycememia. Then you're going to start, as more and more fat is present, accumulating it in the liver, fatty liver. Now the liver can't regulate blood glucose levels. It's going to have insulin resistance in the liver, and you start getting fasting hyperglycemia. Eventually you're accumulating fat in the pancreas, and you start getting damage to the pancreatic beta cells, and they lose their ability to make insulin, and then you get insulin dependent diabetes, and then the other typical complications: retinopathy, hypertension, heart attack, kidney failure. Most of them die from heart attack, most common cause of death, impotence, amputation. So I'm showing this as a way that it's progresses, and it's all just accumulation of more fat and complications related to the fat and to the advanced glycation end products. So there's good news in all this. Eat less fat. Okay. When you got insulin resistance, you have a problem with your fat too. You're going to have, because you have insulin resistance, you can't get the insulin to shut off the breakdown of the fat, the lipolysis. So you keep on pushing more fat into the blood, and you end up in a vicious cycle of you constantly having high blood lipids, and that leading to insulin resistance and all these other complications. And we're going to be talking a lot about this one here, methylglyoxal. That's the big thing to cause advanced glycation end products. The diacylglycerol right here is going to cause, is part of the pathway of insulin resistance. Okay, here is the electron transport chain. This is the inner mitochondrial membrane. So IMM is for inner mitochondrial membrane. Here's complex 1. Pumps out a proton into the intramembranous space. Intramembranous space is between the inner mitochondrial membrane and the outer mitochondrial membrane. This is where a proton gradient is built up inside the mitochondria. Then complex 2 is related to the FADH2 of Krebs cycle. Electrons are given to coenzyme Q. Coenzyme Q passes its electrons to complex 3. Complex 3 also pumps a proton into the intramembranous space. Electrons are handed off to cytochrome C. You don't have to know all these details, but what I'm trying to show you is how these things are coupled together. Electron transport through the electron transport chain used to pump protons into the intramembranous space. A big gradient, like negative 160 millivolts, is built up here, and then that's going to be harvested to take a proton in the opposite direction through ATP synthase to make ATP. This is how electron transport is coupled to oxidative phosphorylation of ADP to make ATP, and that's where the body makes energy. That's where most energy is made in the body. And you can only get about 2 ATPs made by glucose anaerobically from glycolysis, but you can get about 32 some people say 36, it depends on certain things, but just say in the ballpark of 32, so 16 times more energy production when you make ATP with oxygen in the mitochondria. Okay, and we're also going to talk about when you get excessive amounts of fat in here, you overwhelm this gradient and it starts to go backwards. When it starts to go backwards, electron transport, that's a disaster. Coenzyme Q starts dropping more and more electrons into oxygen in the mitochondrial matrix, and those get made into superoxide, and that's a free radical or reactive oxygen species, and that can bounce around like a super ball and start damaging things. We're going to get to that in a moment here. The main focus of this talk is going to be about the advanced glycation end products, but all these things are happening simultaneously. Okay, so normally glycolysis means that glucose goes into the cell. Here's the plasma membrane right here. The glucose is phosphorylated by hexokinase to trap it into the cell. I call it glucose because it's an aldehyde. That'll make it easier to remember in other contexts if we go through the biochemistry. But the bottom line is the phosphorylation traps the glucose in the cell, and it goes through a series of steps, ends up in glyceraldehyde 3-phosphate. This is a real important molecule, glyceraldehyde 3-phosphate. Its enzyme is going to be glyceraldehyde 3-phosphate DH for dehydrogenase. This enzyme is going to be inhibited when there's insulin resistance. I'm telling you guys the inside secrets of diabetes that most endocrinologists don't even know all this stuff. And the reason though, why do you want to know it? Because when you know it, when you get this, what we're going to go through, it becomes so obvious you want to reduce dietary fat. It's not even funny. And understanding will enable you to make wise choices. Okay, so now we've got insulin resistance, backup of electron transport, backup of Krebs cycle, and that's going to lead through inhibition of this enzyme in particular, glyceraldehyde 3-phosphate dehydrogenase. So the molecule, the substrate right before this enzyme, glyceraldehyde 3-phosphate, starts going down side pathways. The glycer is like glycerol, 3 carbons, and that makes diacylglycerol. A triglyceride is a triacylglyceride, so a diacylglyceride just means 2 fatty acids attached to a glycerol. But anyways, that activates protein kinase, which puts a shutdown on the glucose type 4 transporters going to the plasma membrane. So what this all means is it stops insulin from working. Normally, insulin binds to the cell, causes the glucose type 4 transporters to be moved up to the plasma membrane, and then glucose can be brought into the cell, the skeletal muscle cell. But once you got increase in diacylglyceride, DAG, forget it. No more glucose coming into that cell from the insulin. The insulin's not going to work. And now this glyceraldehyde just keeps accumulating and starts getting made into things it should not be made into. It gets converted into methylglyoxal, typically abbreviated MGO. This is actually a big deal. So the excess glyceraldehyde 3-phosphate right here gets made into a side pathway of MGO, methylglyoxal. And that methylglyoxal goes around generating advanced glycation end products all over the place, inside the cell and outside the cell. It's a disaster. Okay, here's just some of the bad things that are happening from the increase in diacylglyceride and activation of protein kinase C. the blood becomes more prothrombotic. And I'll leave this as this because it's not the focus of our talk, but what I'm trying to say is once you unleash the dogs of war with elevated dietary fat and you get a backup of all these metabolic pathways in the cell and you get insulin resistance, all kinds of bad things are happening. The blood becomes more hypercoagulable, your endothelial cells stop working, you have more inflammation, you have immune suppression, you have increased autoimmune disease. Everything bad is what happens from these high-fat diets causing insulin resistance. This is just, I went into some of the more precise mechanisms if anybody wants to look at it. The best paper ever written on diabetes, this one right here, Pathobiology of Diabetes Complications, a Unifying Mechanism, the Banting Lecture 2004, written by Michael Brownlee from the Journal of Diabetes. And you can go to ADA's site and you can watch his video on this. He lectures it. It's great. Shulman's lecture is an awesome lecture. Gerald Shulman, he's the MD, PhD guy from Yale. And he's a genius, and he's the one who figured out with the NMR spectroscopy that the earliest thing detectable is accumulation of fat in the skeletal muscle cell causing insulin resistance. I talk about this more in individual other lectures on this, but for our purposes today, we're going to focus on the AGEs. We talked a little bit about the different types of sugar molecules. Basically, glucose is by far the most important sugar to know about. It's the most common one in the human body. It's the one we need to metabolize. It's the one our brain wants. Fiber is really made out of glucose, but it's connected to each other in a different way such that we don't have access to it. But our gut and our colon bacteria do. Okay, and I talked about this in my last lecture on AGEs. We're moving towards something new. Trust me, I'm going to give you something, a big surprise about AGEs that is you're going to have an AO, academic orgasm, when we get to that. All right, so here is just simplest sugars. We talk about it when the hydroxyl group's on this side, it's a D. The sugars in the body are almost all D. You should just consider them all Ds. That was my mnemonic, DOG, D to remember it, and where the oxygen is of the hydroxyl group in an aldehyde glycer. Okay, sort of set it backwards there as a mnemonic. That's more like the biochemistry of comparing sugars to proteins. All right, here's what's happening. This is an important slide. This actually really matters. Here's a Fisher projection. Looks like a fishing line, a straight open configuration of glucose. Glucose is an aldehyde, so it's got a carbonyl group at the end. There's just going to be a hydrogen coming off here. And this can cyclize, turn into a cyclic ring, and from the fifth carbon to the first carbon. And once that happens, the ring form can be alpha configuration. Alpha is short, so the OH is down. The beta is tall, so with the beta, the OH is high. So OH high on the beta, beta is tall. Alpha is short, so the OH is down. And the point is, it opens up, and then it goes to a ring. Opens up, goes to the other ring. And in this process, while it's briefly opened up, less than 1% of it's open. But in that process where that 1% is open, that can potentially bind to other things. That can glycate, for example, hemoglobin. That's hemoglobin A1C. It can glycate albumin in the blood. It can glycate immune globulins in the blood. It can glycate collagen in the extracellular matrix along the walls of arteries and thicken the basement membrane, capillary basement membrane thickening. It can also damage the kidney filtration system. It can damage the collagen around the spine. It can lead to increased stiffness there, cause wrinkles in the skin, causes all kinds of problems, causes cataracts. These advanced glycation end products are bad. Okay, here is the carbonyl group of the aldehyde on our D-glucose molecule, and it can bind to nitrogen side chain on an amino acid of a protein or to the end amino group. A common one is like lysine. It's got a big nitrogenous side chain on its amino acid. So once this bond forms, it will become irreversible. That's why it's called an end product, advanced glycation end product. And these will stiffen collagen and other proteins and make them dysfunctional. Okay, some of the stuff from the literature. Diabetes and its complications are rapidly becoming the world's most common cause of morbidity and mortality. So what he's basically saying is diabetes is what does the most damage to patients, hypertension and diabetes. And that diabetes, in this author's opinion, is the most common cause of disease and suffering amongst humans in the entire world. And what's it caused? By eating fat. So, because, I see all kinds of nonsense on the internet saying, oh, high-fat diets are good and, get your good fats. All this BS. No, excessive dietary fat. When I say excessive, I'm just thinking, anytime you're getting over 10%, you're looking for trouble. You want to keep it low. So anyways, I like it low, and you could survive and be just fine with only like 2% dietary fat. There's studies where they fed people less than 1%, 0.7% fat, and they were fine. Okay, AGEs, from excessive blood glucose levels, especially it's worse in meat and fried foods. They have the most AGEs. The longer the protein lasts, the more vulnerable it is to glycation significantly. Our body normally has some AGEs, and it's good at clearing them out in small amounts. But when they're in large amounts, they damage, especially these long-lived plasma proteins in the blood. So hemoglobin glycation, because RBCs live about 3 months, it's an indicator of blood glucose levels over the last 3 months, or in diabetic control. Okay, here's what I think is the best slide in this whole talk. This is a diagram. We just talked about methylglyoxal, MGO. So here was that glyceraldehyde 3-phosphate we talked about earlier as being a substrate in the pathway of glycolysis for glucose metabolism. So here was that enzyme we talked about getting inhibited when there's a insulin resistance and a backup of electron transport, backup of Krebs cycle, and then a backup of glycolysis. So when this is inhibited, a side pathway gets activated for glyceraldehyde. It gets made into MGO, methylglyoxal. Okay, now that's a disaster. It's a double carbonyl, meaning there's 2 carbons with double bonds to oxygen, and it's hyperreactive. It starts to glycate, bind irreversibly to proteins in the cell, causing damage to cellular proteins in the cell and dysfunction. Okay, and then here's the kind of stuff I think people are especially interested in. The MGO can go out of the cell and start to glycate. That means they're irreversibly bind to form AGEs, advanced glycation end products, with the collagen in the extracellular matrix. So it's going to stiffen the tissue around these cells. It's also going to damage the capillary basement membrane around your endothelial cells, and that's going to lead to capillary basement membrane thickening, decreased oxygen delivery to the tissues. It's going to damage the basement membranes in the kidneys, lead to decreased ability of the kidney to filter the blood, so kidney failure. The MGOs will form glycation with the plasma protein albumin, which is a carrier protein for a lot of other hormones, for example. And then these AGEs will bind to what's called the RAGE receptor. The RAGE receptor is just a receptor for advanced glycation end product. And then this will activate a pathway, number 1, to produce more inflammatory molecules from nuclear factor kappa B, and you get all these inflammatory cytokines. These inflammatory cytokines are also associated with increased platelet aggregation. When you activate platelets, the activated platelets are more prone to clotting. They're more prone to, it's called aggregation. They also tend to wrap around metastatic cancer cells. They increase your risk of metastatic cancer. Diabetes, increased risk of metastatic cancer. High-fat diet increases metastatic cancer risk. Advanced glycation products activating platelets causes increased cancer risk. That's also why excessive psychological stress or stress related to caffeine and whatnot can increase risk of metastatic cancer, because all these activated platelets, they hide metastatic cancer cells from the immune system. The activation of the RAGE receptor also activates the NADPH oxidase to make more reactive oxygen species, so you get more oxidative stress. So this is the way it tends to be in diabetes. And again, this all arises from eating a high-fat diet in particular. You get all these negative inflammatory pathways going, and they can end up in a vicious cycle of more and more tissue damage. So it's not rocket science. Minimize your dietary fat intake. You don't got to be a genius to figure that out, and it's easy to do. Okay, here's a paper about, review paper about advanced glycation end products. Here's the author's names down here. You can look this one up. Sorry, I kind of cropped out the top name of the paper. So it talks about the things we talked about: glycation, platelet activation, increasing your risk of cancer. Oh, and here's the other one. A lot of people have been interested in, what about osteoporosis? Yes, I talked about that in my recent spine lecture, but here it is in this article. Impairment in collagen alters osteoblast differentiation, the ability to make bone, and you end up with skeletal fragility, osteoporosis. Okay, so whenever you hear somebody telling you to eat more fat, you can know the person's either an ignoramus or a liar. So just be advised, okay? Let's see. Excessive AGEs, they accelerate aging. You'll be more prone to getting wrinkles. Here's a big thing: cataracts. They make you go blind. Okay, they make the blood more prone to clotting due to their interaction with fibrinogen. So that's bad, increasing your risk of atherosclerosis, including blood vessels. So oxygen supply to other tissues. They talk about albumin glycation leading to platelet aggregation. That's bad. And the longer the half-life, the worse. So things with long half-lives, especially, the hemoglobin molecule, about 120 days along with the red blood cell. Also, here's a big thing that people care about these days: the glycated immune globulins, IgG, immune globulin G. it then can become a target itself for autoantibodies. So you're causing autoimmune disease. This contributes to autoimmune disease. We talked about leaky gut as a major cause of autoimmune disease, but high-fat diet does it not just because it causes leaky gut, also because it causes advanced glycation end products that distort the shape of the immune globulins, and the body attacks its own antibodies. So it's an autoantibody. It's a disaster. MGO is considered a major contributor to immune suppression in diabetic patients. And because it's not making, your antibodies are not working because they're getting glycated, the person's also then less able to mount an immune response to an infection. That's partly why diabetics are so much more likely to die from an infection than as an otherwise healthy person. Okay. They talk about collagen being a major target of enzymatic glycation. It's got a lot of nitrogenous side bases, lots of lysines, and that's damaging to your bones, to your blood vessel walls. Collagen is the most common protein in the human body. It also has the longest half-life in the human body. So when you damage your collagen, you're putting long-term damage into the body that structurally makes it weaker and more prone to all kinds of problems. Okay, a little bit about how much AGEs, advanced glycation end products, are found in food. So we talked about, first of all, insulin resistance with backup electron transport, Krebs cycle, and glycolysis forming MGO, methylglyoxal, in the cytoplasm of the cell. So that's a form of endogenous, formed from inside the body. You also get them from hyperglycemia formed in the blood. So intracellular is the MGO, methylglyoxal. In the blood is your standard advanced glycation end products due to high blood glucose levels, hyperglycemia. And then the third type of advanced glycation end products just from what you eat. Like when white bread gets browned, that browning is advanced glycation end products largely forming. And then the thing it turns out that's really bad is the more fat is in the food, the more high protein it is, and the more fried it is, the worse it is, and cooked at high temperatures, prolonged amounts of time. So fried bacon is a disaster. 12,000 advanced glycation end product units, relatively speaking here. So all your animal products tend to be quite high in AGEs. Olive oil is high, mayonnaise too. And just for comparison, look at this. An apple is about 13, okay? White rice, banana, carrots, about 10. So it's orders of magnitude higher with animal foods. Okay, that's an important point. Your body can easily clear out all this stuff. When it gets up into these numbers, you start having tissue damage. And I collected some stuff from the literature I think was kind of interesting. So first of all, advanced glycation end products accumulate in the lens. They cause vision impairment. They cause cataracts. I can tell you almost every single demented guy I see, they always got cataracts. To me, when you start getting these cataracts that require surgery that far along, usually it's an indicator you've lost a lot of brain cells too. All right, now here's something. There's another big thing you see. Oh, nuts are so good. BS. Look at this. Nuts are super fat, like 80 to 90% fat. Look at the AGEs in these things. Of course, it's worse when you cook them. If you roast them, it's really high, 6,600. Look at that compared to, a potato or an apple or some other simple raw plant food. They're bad. Okay, avocados, 1,500 something. Okay, now here's a good one. Look at this butter. Butter is 26,000. Now you got all these lying dumbasses, physicians and PhDs telling people to eat butter on the internet. Look at the amount of AGEs in here, okay? 26,000 from whipped butter. Okay, let's just say regular butter. 23,000. These are off the charts. You want to damage your body, eat this crap. Okay, and the guys saying that, they're either total ignoramuses, which is a lot of them, or they're just blatant liars, which is a lot of them too. That's one of the reasons why I keep making this channel. Even though I don't get hardly any views, I'll tell you the truth. You can read this for yourself in 5 seconds because there's so much crap out there. My friends tell me it's so hard for them to get good information on the internet. Here it is. You can just look this all up yourself. These are the AGEs in these products. Here's the article. You can find them from this and advanced glycation end products. And there's other pictures here I've got where all the reference parts will be on it. Okay, cashews, 6,000. Cashews roasted, 9,000. Okay, peanuts, 8,000. Pine nuts, 11,000. Sunflower seeds, anywhere from 2,500 to 4,700. Walnuts, 7,800. So this is funny. orders and orders of magnitude. These nuts, because they're such a high fat food, and it's much worse if you start cooking them, but they're bad, okay? They're really high in AGEs. I wouldn't eat this stuff. You know, on a holiday, to make somebody happy, would I have, you know, a spoonful of nuts if I had to? Maybe, but that's not a big deal. And I have had Brazil nuts in the past to get selenium, but you can get selenium in other ways. I may talk about selenium in the future. It's a little bit interesting. But anyways, getting back to this, potatoes. If you just eat a boiled potato, hardly anything, 17. But if you start doing other things, like at McDonald's, you fry the potato, then it's got 1,500 AGs. Bad. Okay, if you put butter on it, bad. And so if you just eat the boiled potato, I just eat boiled potatoes normally. They're a great food, one of the best foods in the world. But the more you start messing around with them, you know, like a typical idiot will eat a potato covered in aluminum, so you get aluminum on the potato. Then they're going to put a bunch of butter and sour cream in there and salt. It's no longer a health food. All right, here, just an apple, about 13, a banana, about 9 AGEs, carrots, about 10, okay? So they're really low. Raw plant foods are super low in AGEs. Okay, this is just another side pathway that gets activated. It's called like the polyol pathway, and you make sorbitol off the, once you got to back up glucose. And the important point is you deplete your antioxidants. So this is, again, another reason why diabetics are pretty much screwed when it comes to trying to defend themselves from an infection, because they've glycated their antibodies in their blood, so they're not functioning well. And then their antioxidant system inside their cells is damaged, so it can't function effectively. So to be diabetic is to be unhealthy. Okay. We talked about this, you know, the electron transport chain and how it starts going backwards here. Coenzyme Q starts dropping electrons down to oxygen inside the mitochondrial matrix, producing superoxide and that whole cascade of reactive oxygen species. Okay. Now here is cardiolipin. Cardiolipin is a very common, super common type of fatty acid on the inner mitochondrial membrane. It's a PUFA, polyunsaturated fatty acid, meaning that it's got extra double bonds. The more double bonds a fatty acid has, the more prone it is to lipid peroxidation. So the point I'm making is when you start eating this fat, you're going to start dropping more electrons out of your coenzyme Q down onto your mitochondrial matrix, making superoxide anions, and you're going to get more and more damage, lipid peroxidation of these cardiolipins. So what I'm saying is you're damaging your mitochondria. If you want to have good energy, you want healthy, happy mitochondria. You don't want lipid peroxidation damaging your mitochondria. So this is another thing that high-fat diets do. They damage your mitochondria, so they drop your energy levels. It's pretty common when somebody goes vegan after being a high-fat eater, let's say in their 30s and 40s, that they get more energy and start wanting to run, you know, 10Ks, marathons, etc., triathlons. Okay, here's a PUFA, polyunsaturated fatty acid, meaning more than one double bond. The carbon in the middle is a methylene bridge carbon, and it doesn't have that tight a grip on its hydrogen. That can get plucked off, and when it's plucked off, there's an electron sitting there. That electron is then vulnerable to reacting with oxygen in the adjacent area, and it'll produce what's called a peroxide. Peroxide is just two oxygens next to each other, and this will often be a free radical with an extra unpaired electron in its outer orbital, which is also hyperreactive, and it'll create a chain reaction, reacting with other plasma membrane and other fatty acids, causing damage to them, and it can damage many of them. And so what's happening is you're trashing your plasma membranes, you're trashing your mitochondrial membranes, you're doing a lot of tissue damage. You know, and the body repairs it as well as it can, but the body repair systems can be overwhelmed. Let's see, what was in here? Oh, this is this guy, Roy Taylor. He's this brilliant endocrinologist, expert on diabetes from England. He worked for a while with Gerald Sheldman over at Yale and the NMR research on insulin resistance. And so he talks about how the great thing about the low-fat vegan diet is that it's easy to maintain long-term. When people try paleo, low-carb, Atkins, and all that, they usually can't sustain it for a long amount of time, and they start eating a half-assed SAD diet quite often. Whereas low-fat vegan, it's easy to sustain it for years because it makes you feel good, and it's easy to eat, and it's pleasant. So anyways, he's good. Okay. Let's see. Oh, Michael Brownlee, he's the genius that wrote that one paper about unifying theory of diabetes complications. Best paper ever written on diabetes. And here's what he says. Almost all diabetic patients die from coronary artery disease. Yeah, it's sad. They're screwed. Okay, and I know most diabetics I talk to over 50, they're cognitively impaired and mentally slow. I feel sorry for them. They're real pathetic. You know, you try talking to them, they're like, oh, I'm doing okay. You know, they're a disaster. And then I'll see them a year later getting their foot chopped off, and they're a disaster. Okay, we're going to talk about the cells that have glucose 1 type transporters in their plasma membrane. Oh, just briefly, some of the things that cause insulin resistance. So excessive dietary fat is by far the most important cause. There's other less important contributions to insulin resistance. Excessive dietary sodium ends up contributing to it, which typically comes from meat and processed food. Lack of dietary potassium and magnesium, that comes from not eating enough plant foods. That's where they are. Excessive stress, because it increases cortisol and catecholamines. They're going to drive up your blood glucose, drive up your blood lipids. Stressful equivalents, caffeine, sleep deprivation, loud noise, strobe lights, anything that stresses out the person. Corticosteroid medications, lack of exercise. Exercise does the same thing as insulin in a sense. It gets the glucose type 4 transporters to go from the center of the cell up to the plasma membrane to allow more glucose into the skeletal muscle cell. So it's really good for diabetics to exercise. It's good for everybody. Obesity leads to chronic increased blood lipids, which causes insulin resistance. High protein diet, we talked about problems related to that. That causes hyperlipidemia as well. I learned that first from T. Colin Campbell in a China study. AGEs, you know, as you get older than 45, you're more prone to these problems. You know, the body gets more fragile as you get older. So watch out for hypothyroidism too. Makes all this stuff worse. Okay, we talked before about, you know, it's been known for a long time, high-fat diet is what causes the main cause of insulin resistance. There's something interesting here. This is NP is for Nathan Pritikin. I was reading through some of his work, and he was talking about, in his opinion, the risk of cancer when you eat a low-fat diet is reduced by 90%. That's an extraordinary number, by the way. Low-fat diet decreases the risk of cancer by 90%, according to Nathan Pritikin. I mean, that is so extraordinary. I'll say it one more time. Low-fat diet decreases the risk of cancer by 90%. Lots of people are sad, you know, and cancer can be kind of a miserable disease. And the point is, you know, here you've got this way to have a great chance to avoid it. Not guaranteed, but it's good odds. Okay, Nathan Pritikin also said 85% of his patients came off all their blood pressure meds. 50% came off of their insulin, off all their insulin of his type 2 diabetics. Nathan Pritikin, there's two main types of diets in the world, low-fat diets and high-fat diets. Okay, there's more to it than that, of course, but that's a good general concept there. Okay, fat causes insulin resistance. Yeah, we know all this, the Sweeney paper, the Rabinowitz paper, the Himsworth paper. So if anybody wants references, here's a bunch of references about high-fat diets causing type 2 diabetes. Lesson learned, minimize dietary fat. Okay. Okay, now we're getting into sort of a new topic related to, this is a toxic aldehyde, hydroxynonenal, and here's the structure of it. This is more something that especially comes up in the context of eating dietary oils. All these omega-6 oils and cooking oils, they're a disaster. You don't want to eat that stuff. Don't ever eat oil. And the big guy who figured out a lot of this stuff was Tetsumori Yamashima. Here he is. Dr. Tetsumori Yamashima, he calls it the toxic aldehyde theory. And he was trying to figure out, he's a neuroscientist, he was trying to figure out why are so many Japanese people becoming demented these days. And it's because they're eating all these cooking oils. They used to be super healthy. Japanese are having more and more health problems. And he also felt that the excess dietary PUFAs, omega-6 especially, were leading to not just dementia, they were also leading to damage of beta cells and increased risk of type 2 diabetes. Let's see. All right. Oh, yeah. And then I made up my law in terms of, you know, what happens. I said the more profitable it is to manage a disease, the less likely a cure will ever be discovered. The more profitable it is to manage a disease, the less likely a cure will ever be discovered. Okay. If you put a type 2 diabetic on a vegan diet within 4 years or less, you can cure, according to Roy Taylor, like all of them if they're willing to make the change. Most people aren't, but it's nice to know that there is an option. Okay, this is just showing hydroxynonenal inhibits ATP synthase, so it's going to decrease energy production in the mitochondria. Here's the reference for this paper. So it's a mitochondrial toxin, this toxic aldehyde related to eating omega-6 fatty acids. It's especially bad in people who can't process alcohol, like that Asians who don't have acetaldehyde dehydrogenase enzyme in adequate amounts, so they get flushing when they drink alcohol. They also are less able to process the aldehyde end products of eating omega-6 cooking oils. So here he is, Tetsumori Yamashima. Hydroxynonenal causes Langerhans cell degeneration in the pancreas of Japanese macaques. That's a type of monkey. So yeah, he is also using this to explain why there's increased incidence of diabetes. And I talked about this before in a previous lecture about why do persons of Indian descent seem to have more diabetes, and I thought this might be the reason. They tend to eat a lot of fried food, and the hydroxynonenal toxic aldehyde byproducts of that appear to damage the beta cells of the pancreas, according to the extensive research of Tetsumori Yamashima. So here's just some papers and references. But it also occur in animals that don't have acetaldehyde dehydrogenase deficiency, the pancreas problems. Some of the brain stuff is especially prone in the ones that have that the alcohol flushing syndrome. Okay, and he goes through this whole mechanism of heat shock proteins. I talked about this more in a previous lecture, but the heat shock proteins are important in the brain cells, especially in your neurons, to recycle dysfunctional proteins. And when you have high amounts of these toxic aldehydes like hydroxynonenal, you're going to end up damaging your lysosomes. And so we're not going to get in that too much. I talked about that in a previous lecture. I'll just briefly show you a picture of it. So here's the heat shock proteins. They're chaperones to take dysfunctional proteins to the lysosome. The lysosome will then degrade these proteins. That's normal cellular physiology in your brain neurons. The heat shock protein, HSP, also stabilizes the membranes of the lysosomes. However, when you eat excessive amount of cooking oils, you'll start to have problems whereby you'll get increased degradation of these heat shock proteins. You're going to have increased activation of this calpain protease. And when it cleaves the heat shock protein, they no longer can do their job to improve lysosomal function. And lysosome enzymes, this membrane will become loose and your cell will auto-digest itself. Other things make that worse, like if there's increased psychological stress, increased dietary MSG, MFG, the monosodium glutamate, manufactured-free glutamate, you'll get increased cellular calcium. So all these things are ramping up the cell. It's a form of excitotoxicity, coupled with this calpain effect and the destruction of HSP. The bottom line is high dietary sodium is bad. All these stimulant-like molecules, caffeine, they're bad. They all make this excitotoxicity worse, and they all predispose to more of this, especially in the context of eating a lot of omega-6 cooking oils. Okay, so what can you do about this? Minimize your dietary fat, minimize your dietary sodium. Fruits and vegetables are great. Try not to drink sweetened beverages to the extent you can, other than maybe just before a workout. All right, well, anyways, that was the key point. So what was interesting about that? I thought what was interesting about it was this idea of, you want to minimize your dietary fat. The more dietary fat you eat, the more insulin resistance you're going to have, the more advanced glycation end products you're going to have, the more damage you're going to have tissues all over your body. So that was the big thing to learn. There's really good reasons why you want to minimize dietary fat, to minimize your insulin resistance and your advanced glycation end products.